Buenas tardes, bienvenidos a todos a esta serie de seminarios llamados Los ecólogos de Krebs sobre la regulación de poblaciones. Esta tarde estamos presentando el quinto seminario. Nos acompaña el doctor Rudy Boonstra, quien es profesor emérito en el Departamento de Ciencias Biológicas de la Universidad de Toronto, Scarborough, en Canadá. Rudy obtuvo su doctorado en UBC bajo Charlie Krebs y Dennis Chitty. Y ustedes ya conocen a Charlie Krebs por las charlas anteriores. Y bueno, también a mí me tocó conocer a Dennis Chitty. Fuimos muy afortunados. Rudy fue director del Centro de Neurobiología del Estrés en la Universidad de Toronto y ahí también enseña ecología de poblaciones avanzadas y las consecuencias biológicas del cambio climático. Rudy ha publicado más de 200 artículos científicos y su trabajo actualmente se centra en la ecología, en la neurociencia y en la epigenética. Él está interesado en el papel del estrés, del eje del estrés, como él lo llama, en las poblaciones, en la regulación de poblaciones naturales. Alguna vez Charlie Krebs me dijo que no estaba seguro si había vida inteligente al este de las montañas rocosas, pero como vamos a ver el día de hoy, sí hay gente inteligente al este de las montañas rocosas. Well, I'm really pleased to continue with this series of seminars. This is the fifth seminar of the series Krebs Ecologist on Population Regulation. And today we have Rudy Boonstra, Professor Emeritus from the Department of Biological Science of the University of Toronto, Scarborough, in Canada. Rudy obtained his PhD at UBC under Charlie Krebs and Dr. Dennis Chitty. He has been director of the Center of Neurobiology of Stress at the University of Toronto, and he also teaches their advanced population ecology and biological consequences of climate change. He has over 200 publications, and his current work includes ecology, neurosciences, and epigenetics. He's interested in the role of the stress axis in natural population regulation. Charlie Krebs used to tell me that he was not sure if there was intelligent life east of the Rockies. But as we will see today, there is intelligent life east of the Rockies. I think he was referring mostly to politicians, not to scientists. Welcome, Rudy. Thank you very much for being with us. The screen is yours.
Okay, it's evening here in Toronto. We were just in the middle of a snowstorm. So I'm going to be talking about population regulation and cycles, a window into the natural processes, which I think is going on there. And it's extremely difficult to, it's been almost a lifetime working on cycles and population regulation. And I think now we're coming to the culmination of that in terms of trying to, getting the evidence that there's something major going on. Okay, so all science is either physics or stamp collecting. Ernest Rutherford made this statement. Ernest Rutherford was the father of nuclear physics, and he actually got a PhD, not a PhD, he did his Nobel work at McGill University. So he has a long history of Canada, and he then went to Manchester and Cambridge. But the question is that right? Is science, is the only science that people do physics, and the rest is essentially stamp collecting? And I reject that categorically. What it implies is that only physics has rules, but no other science does. So the question then is, what are the overarching rules or paradigms which govern our efforts in biology and in ecology? So what you see in front of you is two stamps. The stamp on the right side is the oldest stamp. It's called a black penny stamp from about the 1840s. It's Queen Victoria. Okay, and on the other side, you see Shania Twain, a Canadian stamp. But if you ask the question, is the only common thing between these two stamps the glue between them, or is there something more fundamental that links these two stamps? If you look at these two pictures, you see a ground squirrel on one side and an old man with a child on the other. And again, the question is there a commonality here? And I think one of the big questions is what controls senescence in animals? And it's sort of shown here between these two. But the other major question is what is the role of stress in animal populations? And both of those questions have intrigued me since I was an undergraduate at the University of Calgary. Okay, so Dennis Chitty has written, he didn't write a huge number of papers, but he wrote a number of very seminal ones. And his first major one that hit me was his 1960 paper. And he made the following statement in his abstract. He said, no population continues to increase indefinitely. And the problem is to find out what prevents this. And that's what I've struggled to do over my career. If you look at the human population where we sit right now, we're looking right now at about 7.8 billion people. When my father was born, just after 1900, there was about 1.5 of us. And ultimately, from our perspective as humans, is this sustainable? Where are we going? Can we continue to increase like this? What will put the brakes on? So it's a fundamental question both for animal populations, all animal, but also for the human population. Okay, so if we take a look at the population processes that control animal populations in the natural world, you can see in black or sort of blackish blue, there are two major categories. There are those you can refer to as the abiotic factors, the physical and chemical factors. And in that group, you could list that could be moisture or temperature, cold, light, fire, salinity, especially in Canada, you know, in the Arctic, it's very, very cold. Could that control animal populations up there? The alternative is the biotic factors, interactions between animals themselves, competition, top-down regulation by their predators, or the other types of predators are the diseases and parasites. Could they all act, or independently, or one at a time, act to regulate animal populations, to essentially limit their abundance? So what's the explanation? What are the processes? Okay, simplistically, you can look at population size as being affected by four major parameters. And we'll essentially exclude the two ones on the side, movement into the population and movement out. And so if we reduce population size to what goes on in terms of reproduction, coming in, and what leaves it according to death, what are those processes? And if we understand them, can we understand the population regulation? So that's been one of my goals. If we look again at these factors, you can call these factors which are external to the population, and that's what we've been doing in the previous two slides ago. Extrinsic factors would be the predators, food supply, parasites, disease, and shelter. All of these could affect the population, but they don't act all by themselves. The populations themselves have internal processes. Sex may be important, how old the population is. The physiology, stress physiology, and other things are really important. Their behavior and their genetics. But I think there's a third factor, or another, a sixth factor, which we haven't really thought about or considered, except for about the last 20 years. Okay, so what I'm going to do in my talk is identify a key problem that I think we have to solve to try to understand population cycles. Before I do that, I'll talk about the key features of the pattern. We'll look at the evidence, and then I'll propose a hypothesis that the things that we've been missing, and it's not entirely missing at all. Dennis Chitty thought about it over 50 years ago, actually almost 70 years ago. What's the role of maternal effects? What he didn't think about was the role of epigenetics, because he didn't know about that. Okay, so as you see, I've expanded the intrinsic factors, and I've included epigenetics at the bottom as a key process that we need to understand if we're going to understand population regulation. And hopefully by the end of the talk, you can see if I've convinced you that in fact it's worthwhile considering, and more than worthwhile. It essentially gives deep insight into what we've been observing. Okay, so let's take a broad brush look at cycles. We'll first look at voles and lemmings and then snowshoe hares. Here you can see at least 5 different studies showing that in fact there are cycles in voles and lemmings all the way from Finland to Norway, Nunavut in Canada, Russia and the USA. And here you see a really nice picture of a lemming in the lower left. Okay, so we know about these cycles. This just shows our evidence. Charlie Krebs showed this slightly different version of this from the Yukon. And as you can see at the very bottom, there's a 45-year period where we have monitored. He started it back in 1973, as I recall. We got back there to 87. And you can see that the arrows at the top indicate in this red-backed vole. This is our northern red-backed vole. And this particular vole species is essentially a major vole all the way across Canada, north of 60 degrees latitude, all the way to Finland, throughout Russian Siberia, all the way to Finland. So it's over that entire huge area. But you can see in our population, about every 4 to 5 years, you get a peak and the populations go down, and then another peak occurs about 4 or 5 years later. Okay, so then the question is what causes population change in these animals? Let's see what's happening. Oh, I made a mistake. I hit the wrong button. Sorry. Okay, so many of our colleagues in the US, in Northern Europe, have favored the role of predation through weasels and other mammals, or the role of food. Are these the drivers of population cycles in these small mammals? And these are 3 papers which show that these processes, these extrinsic processes, may be sufficient, but these papers show that they're not necessary. These small mammals will continue to cycle. So there's something in them over and above these extrinsic factors which is driving them. And then the question is, what's the mechanism? What mechanism is both necessary and sufficient? We'll go over very briefly to the snowshoe hare cycle. Stan Booten showed this slide, and you can see that they go through cycles of about 9 to 11 years. It's very characteristic, and we're still continuing to study them in the southern Yukon. Okay, so the cause, and there's a series of papers listed here, but the cause of the snowshoe hare cycle appears to be top-down. It's predation, not food, is the dominant mechanism. It acts both through direct mortality, where particularly during the decline, but even before, from our radiotelemetry data, between 80 and 90% of the hares are killed by their predators, those that die. But over and above that, there's another factor going on, and that's the indirect stress effects that affect the hares. They know that the world is turned against them, and the question is, are they stressed by the thought, the threat, however they're incorporating it, of dying? And our evidence of the previous 2 cycles is definitely the indirect effects are also playing a major role to cause the decline and then the low phase. Okay, so the real enigma, or the problem, the real problem in these cycles is to understand what causes the decline, and then the low which follows the decline. What is going on? And I think this is the heart of the issue with respect to understanding population cycles. You can see a paper down below there where we say lay out these problems and identify that I think the low and the decline is the central thing that we must solve if we're to understand how these cycles function. Okay, this just shows you some of my data from Southern Ontario. If you look in 1978, the bars indicate when they're not breeding, and the open areas indicate when these meadow voles, and the meadow voles basically have a huge distributional range. They occur all the way from northern Georgia all the way to Alaska. So they basically occupy much of the grasslands and meadows of the top half of the North American continent. Okay. I started studying them when I came to Toronto back in '78, and everything was going perfectly until about January. We got 2 intense bouts of freezing rain. We're sort of on the cusp of southern air masses and northern air masses. And after that freezing rain bout in February of '79, I could find dead animals all over the place. The populations took a nosedive, and then they went through, and we didn't, that freezing rain bout was not repeated. Then we can see that they went through a very nice cycle, an increase phase, breeding like mad, a very short non-breeding period during the next winter. The populations had increased. They showed a slight spring decline and then a massive increase to a peak in the summer of '80. And a long non-breeding period. And here's where it hits the road. The populations collapse. They not collapse, they drop drastically. But the next summer is the problem. And this is replicated in Charlie Krebs's work. It's seen in others. They have basically the same number of females that are breeding in both summers, but they're not producing babies. And this is an enigma. Okay, this just shows it here. Index of early juvenile survival, good numbers of babies produced per pregnancy, and the increase in peak and the decline, they're doing extremely poorly. The population doesn't go anywhere. And so I think is a good demonstration of the problem that we're trying to solve. Okay, it's echoed by other work that my colleague Steve Mihawk and I did. What he did, he worked in Manitoba, which is northwest of where I'm right now. And he brought in animals from 2 phases, from the decline phase and from the increase phase. And you can see it there. The increase phase are in red, the decline phase in blue. And essentially, the voles taken from the increase phase breed well in captivity. And remember, the animals in captivity have the best of all possible worlds. They have no competition with other animals. They have all sorts of food and no predators. Nevertheless, those from the decline don't do well. So in other words, there's something intrinsic in the animals that when you bring them in, they either do well or badly based on what they were doing in the field. If we turn our attention to what's happened in the Snowshoe Hares, the place where we worked is the Kelowna field site. You've been introduced to that site if you've looked at the other lectures. It's up there. It's almost 6,000 kilometers by road from where I live. It takes me about 24 hours to get there when I go and work in the Kelowna site. But this just shows you the typical snowshoe hare cycle and lynx. But I want to point out this low phase again. You see the double arrows, and the low phase varies from cycle to cycle. You can see it being long between the cycle from 1980 to 1990. It's shorter the next cycle. It's fairly long in the third cycle there. So that's the first thing to note from here. The other thing to note is that they too, the snowshoe hares, show this alteration in reproductive success. During the increase phase, they have 4 litters. During the decline phase and low, at least the early low, they only have 2. And so the question is again, similar to what, not identical, but it's similar to what happens in the vole and lemming cycles. So what's going on? This shows similar data from Lloyd Keith Alberta, he was one of the first persons to look, not only the first, there are others, but look at hares, snowshoe hares cycles in Alberta. And if you look at the red line, you can see that the reproductive output starts declining even as their animals are going into the increase, and they really bottom out during the decline phase. So it's the same basic phenomena that we see in the Yukon is also seen in Alberta. And again, if you bring snowshoe hares from these different phases, from the peak phase and from the low phase to the laboratory, give them the best of all possible worlds, they maintain this sort of intrinsic difference between animals coming from the peak and the low. Hares taken from the low phase breed well in captivity, those from the peak don't. And so again, I'm driving home the message. There's something fundamental about these animals, which was programmed into them in the field. And there's nothing you can do when you bring them into the lab about that. Okay. So as a summary, in these examples, demography during the low appears to be a delayed consequence of factors acting at an earlier time. And I suggest, I believe, that in the voles, the memory of the past may be related to the social density and competition during the peak phase. It's what's going on then, which essentially programs them for the future. In hares, that memory is related to the increasing predation risk that they're experiencing during the peak and the intense predation risk during the decline. So the factors which are affecting them are different, but I think the consequences internally are similar. Okay, so this is my human angle, just to bring you up to steam that you're not exempt from this. If we look at humans, but also the animals that I'm studying, stress is a problem for all of us, particularly now, but it's always been there, all right? You don't get out of life without having periods of intense stress. So this is a nice picture that was put in my university magazine. If you take a look at the picture itself, you can see that these two humans, she's already got a face mask on. And that was probably about 7 or 8 years ago when this picture was drawn. It was sort of thinking about the future. But if you look at that picture, up in the, you see a meteorite about to hit the Yucatan Peninsula. That's a sort of an unknown problem. Floods obviously can be a major problem. Artificial intelligence is coming to be a major problem, and I suspect it will intensify. Nuclear war in the lower left corner, and in the really lower left, you can see dictatorships like Hitler, etc. Okay, so if we look at the text, as I say there, humans have a lot to be worried about. They worry about their family. They worry about putting food on the table, about their job, whether it's going to disappear because of artificial intelligence. And all of us worry about dying, I think. We also worry about disease. COVID-19 is an obvious factor, but one only has to think approximately 100 years ago, Spanish influenza, it appears, evidence indicates it affected about 500 million people, and between 50 and 100 million people died. There's a really good book on this by Laura Spinney called Pale Rider, a very good overview. But politics and power, climate change, war, etc., economic collapse, all of these things are things that we can worry about, and some worry about it excessively, but nevertheless, it's always on the back burner. Okay, just another example. If you lose control, or if you're that point in society which doesn't have much control, your probability of dying is higher than if you have a lot of control. This just shows you a very nice study looking at the probability of dying of British civil servants. So you may think, well, that's going to be a very cushy job, a nice thing to do, but that's not so. If you're in administration or you're a professional executive, you are at the top. You're in control of everybody else. The alternative is if you're in the clerical staff, or if you're doing physical work in the British civil servant, you're at the very bottom. So if you look at the sort of average death rate, it's 1. If you're in the first 2 categories, you do much better. Your probability of dying is much less than if you're in the other 2 categories. And this occurs all the way through from 40 onwards. You can see that the data is split up first into 15-year intervals, then a 4-year interval, and then the last period of life. But not having control stresses you and has direct consequences on, say, the number 1 problem of dying, which is a heart attack or cardiac problems. Okay, so I'm going to argue that maternal effects and programming we need to understand how that works if we're going to understand these cycles. So maternal effects occur when the maternal phenotype affects the phenotype of her offspring, in addition to her direct genetic contribution. So she passes on to her babies her milk. She passes on signals when she's pregnant in utero, and these can all affect the baby's fitness over and above her genetic contribution. So this is just sort of a summary of how maternal effects can operate. You have an environmental signal, whatever it is, can affect the individual, the cohort, or the entire population. And I would argue that in the cyclics populations, it affects the entire population. It affects early development at some point, affecting survival and reproduction, and ultimately it affects lifetime reproductive success and fitness. Okay, so let me give you a couple of examples to show that in fact it's real and that it has major consequences in both the laboratory and in humans. And then I'll walk through some recent evidence that we have in voles. Okay, so first, before I go through that, let me introduce the whole topic of epigenetics. Okay, so epigenetics is the changes, the study of the changes in gene activity that are not caused by changes in the DNA sequence. In other words, the DNA remains the same, the sequence remains the same, but in essence, what changes is whether those genes, the genes in the DNA, are expressed. They may be upregulated or downregulated because of epigenetic changes. So changes in gene expression result or can result in changes in the phenotype. And such changes can be caused by changes in stress. It can be caused by changes in nutrition. For example, in humans, periods of hunger. Similar things could happen in animal populations, or other environmental influences that the animal experiences, and in humans, which are caused by various forms of adversity, such I listed. Okay, epigenetic modification can be caused by a number of things. It just shows you a chromosome there. I'm only listing 2 types of epigenetic modification. The one I'm going to focus on is DNA methylation. So that's the very bottom one. It essentially acts to inhibit gene expression. The other one, there's a number of others. Another one is histone acetylation, which promotes gene expression. But what I'm going to focus on in this talk is DNA methylation inhibiting gene expression. Okay, so here's a laboratory example that I mentioned. This is, when I first read this, it's utterly astounding. It seems like I'm the most trivial thing to look at, and yet the consequences epigenetically are profound. Rats, this is obviously a lab rat. Lab rats, when they have the females, have babies, you could split them up into lab rats that are really good mothers and those that are poor. Good mothers lick and groom their babies a lot. Poor mothers don't. All right, they don't do that. And that just shows you the percent of the animals that lick and groom. Okay, so just step back for a second. This is a shot which shows the stress axis. And this is a schematic, but we know a great deal about how the stress axis functions. At the very top, you have essentially the highest level of the stress axis called the hippocampus. And the hippocampus is sort of the master director of stuff which goes on below it. And that is, the key elements of that are what's called the glucocorticoid receptors. You've got two receptors there, one in orange called the mineralocorticoid receptors, and they are only found at this level only in the hippocampus. They're not found in the hypothalamus, which is the next level down, or in the pituitary or the adrenals. The other one, and the MR, the mineralocorticoid, determines sort of the basal levels of the key hormone, which is a glucocorticoid, cortisol, and many of us, corticosterone, things like the rodents. It determines sort of the basal levels. But then over and above that is what's referred to as the glucocorticoid receptors, the GRs. And they can vary depending on the animal's history, the severity of the stressor, whether it's chronic or not. They can vary, and that basically changes everything downstream. So again, if you follow the downstream, you've got the hippocampus at the very top, then the hypothalamus. I've got a note there called PVN, the paraventricular nucleus. You can see the GR is there. Then you have the pituitary. The pituitary essentially releases all sorts of hormones, one of which activates the adrenal cortex to kick out the thing in black at the bottom called the glucocorticoids. Now again, remember this stress axis, or this axis, I should say, operates all the time. It operates not just in response to stress, but every day it has major functions affecting energy levels in the body. And so you can see at the very bottom, it affects mobilization of energy. If there's high levels, it can suppress growth, etc. So normally when an animal's in good condition and is not stressed chronically, the glucocorticoids are extremely hot. They have many effects, and there's rapid feedback to the level of brain to essentially shut it down. When animals are chronically stressed, like when the snowshoe hares are chronically stressed by predation, then the feedback becomes less intense. It's compromised. And so the signal on the right side, the thickness of the signal, is not as effective. The feedback loops are less pronounced. So the stress axis in mammals is absolutely critical. Okay, so what I'm going to show you now is how this works out in these rats. Okay, and so you've got two groups of rats. Remember, there's rats which lick a lot and groom a lot, and those which don't. The high-licking, grooming animals are good mothers, and the low ones are poor mothers. And what my colleagues, and again, this is work from McGill University by Michael Meany and Moshe Schiff. It was absolutely amazing work when I first saw it. I first saw it in 2004, but I've been following this literature, again, puzzling about maternal effects. And so at the very top, you see that pups that are receiving less licking and grooming by the mother has major effects on, as you can see, that it's called the glucocorticoid receptor promoter 1,7. Okay, so this is not going to mean anything to you until I explain it, but it's the 1,7 promoter, which is methylated. Okay, and what it does is this promoter, these series of blue lines, but it's only the 1,7 which is important. These blue lines affect whether downstream the gene which makes GR is actually expressed. So as I say here, this results in fewer GR receptors being made. So if these are methylated, fewer GR receptors are made during development of the pup, and as an adult, the adult rat can't handle the stress as well as pups from high-licking grooming mothers. Okay, so again, going back, how the stress axis is organized is absolutely critical in dealing with stress, all right? It acts normally, but if it's compromised by even such as a simple thing as a mom who either licks you or doesn't, it directly affects the stress axis. So what that shows us here is that you've got high licking grooming mothers where the promoter, this I7, is not methylated, and it produces lots of GRs. That's on the essentially the right side. You see the hippocampus there. There's lots of GRs. So it's got a very tight system of regulating its stress axis, a highly functional one. The alternative is on the other side, again, the hippocampus, but because of the methylation of the promoter, the gene is not expressed and it makes relatively few glucocorticoid receptors. So its stress axis is not functioning well in comparison to the other ones. Okay, so this looks like a trivial example, but it was absolutely foundational. If you look at the bottom, you can see the reference there by Weaver et al. And the key people are at the end. Weaver is important as well, Ian. But you can see that since 2004, this paper has been cited 6,176 times as of today. So it's absolutely regarded as a foundational paper in terms of maternal programming and epigenetics. Okay, let's turn our attention to a human example. And so you can see, now you can see where the rubber hits the road. How does it affect us? It's all very well to affect rats and maybe voles and lemmings, but does it affect me? Okay, so the human example looks at the impact of childhood abuse on essentially the stress axis, on the glucocorticoid receptors in the hippocampus. And again, at the very bottom, I should have deleted this, but you can see that it was published essentially 5 years after the previous one. And it's been cited almost 3,400 times. Okay, so what did they do? Here's the title of it. Childhood adversity alters the stress response and increases the risk of suicide in later life. The work again took place at McGill, and Quebec has a brain bank of human brains. So what the scientists did, McGowan, Moshe Schiff, and others, what they did is they accessed the brain bank to look at the brains and the hippocampus of people who had died. And the people who had died between 30 and 40 were men. Let's see. Okay, so there were 3 groups. The first group had committed suicide between the ages of 30 and 40. And when they look back at the history, these men had been severely abused as children. The other group also committed suicide, but they were not abused. There was no evidence of abuse in the second group. The third group were the controls. They had died of other causes. They may have stepped out onto a crosswalk and been hit by a car, but essentially their past did not determine their cause of death. They died of other causes. Okay, and so they did a complete search of something like 7,000 base pairs. So if you look at the human suicide, and that's on the right side, look at the DNA methylation patterns and the genes which are showing that. It's not just the GR methylations. There's a large number which are affected. And you can see those on the right, and on the left are the non-abused controls. They're fundamentally different. Essentially what they found is that the controls and the men who had committed suicide, which were not abused, were similar. Non-abused controls and suicides without abuse were the same. The other men were fundamentally different in the way their brain was organized. So, you may have heard the term, you've now got a good life, get over it. But imagine if your brain is programmed and you cannot get over it. And that's what this data indicates. If you look at rats and their maternal care, it's high maternal care on the left and low maternal care on the right. And again, these blue squares indicate the areas where the problems are in the genes in either the rats with low licking mothers or the human abused. Okay, so the point here then is that the thing which we are studying is not limited. There's evidence to indicate that we should be looking at this in natural populations as well. Is epigenetics through maternal effects playing a role? Okay. Okay, now let's turn to the last point of my talk, which is the cycles and voles and lemmings. I'm not going to talk about snowshoe hares. And there's a real problem, just to bring you up to steam. If you're looking at humans or rats, we now know the entire genome of both of these. So for humans, in 2001, they had sequenced two groups, a government group and a private group, had sequenced the 3 billion base pairs of humans. Rats obviously are a major model, so we know them intimately. When we're trying to do the same types of things with cyclic voles and lemmings and snowshoe hares, we have a problem. In other words, many of these are the gene sequences are not well known, so we either have to sequence them or go to other rodents to try to understand the role of methylation there. Anyways, so here's the I think a key issue with respect to voles and lemmings. During the breeding season, females are highly territorial. What you see on the right are meadow voles. This is from Dale Madison's work. And you see that they basically don't overlap. These are 6 different females and they don't overlap. They're highly territorial. And the work of this type of, this has been observed in many studies. On the left are Townsend's vole. This is the work from Xavier Lambin. Again, it echoes the same thing, that females are extremely protective of land, and that's where they have their babies. So then the question is, does that competition for space intensify as population density increases? And does that have ramifications? So you could say that as a hypothesis. Females at the low have lots of space and lots of babies. Females at the peak are protecting their property and they're competing for land, and that in itself could either distress the females in utero or post utero, affecting their babies. Okay, so if we summarize the demography of peak and low animals from the low, during the peak, I've indicated this already, but the breeding season is shorter during the peak. Young females delay maturation often until the next breeding season, so they have to wait an entire breeding season because they cannot find a place to live. There's a lower percentage of these females who are reproductive, and they often have very poor survival. Juvenile survival, in the low, the breeding season is much longer, they mature early, more of them are reproductive, and they survive well, as I indicated in the data from Ontario. Okay, so Dennis Chitty, he's on my right. He was my supervisor. Charlie was my mentor, but he wasn't my supervisor. This is a picture from 1984, when obviously I was much younger, and I had just come back from Australia. In Australia, Charlie helped me get me there, and I did something which was very important for me. I learned how to measure hormones, endocrinology, stress, etc. And we were just passing through, and we went to visit Dennis and his family, and we visited Charlie as well. Dennis proposed what's on the left in 1967. He was trying to understand all his work that he had done at Oxford from the 1930s to the 50s, trying to understand, and he had excluded the extrinsic factors as factors to explain population cycles. His only explanation was that there were intrinsic differences between animals at the different phases. At high density, they were selected for growth and aggression. And at low density, they were selected for high reproduction and maternal care. The mechanism driving these cycles, he postulated, were these genetic differences, fundamental differences. So his mechanism was Mendelian selection. Okay, I was a student of Dennis's, and he proposed this in 1967. I worked on that when I came to Toronto on meadow voles, and then we did it again in lemmings. Both of them rejected his hypothesis. In other words, Mendelian genetics, we did breeding experiments, one father, two females, and so on. We worked heavily on this, Boonstra and Bogue and Boonstra and Hachachka, and in both cases, we rejected the Mendelian genetic explanation. So he was wrong in the mechanism, but was he right? Was there, did he have insights that maternal effects were occurring? Because he didn't know. So ratchet forward 50 years, and now we're recognizing the importance of epigenetics, something that Dennis didn't have any idea about. Okay, so our hypothesis was that the social environment with high population density can drive selection for a low reproduction, epigenetically produced phenotype, which results in population self-regulation as an intrinsic mechanism driving these cycles. Experiment, simple. Well, it wasn't simple, it was a lot of hard work. Create high and low density meadow vole populations in spring, sort of replicating peak and low phase, allow the females, or increased phase, allow the females to breed and examine the epigenetic changes in key endocrine genes involved in reproduction in the offspring in late summer. Okay, so here's what we're postulating. Epigenetic alteration of endocrine traits. Again, the key problem is one of spacing behavior. And so here you have a vole, either at high density or low density. And we hypothesized that at high density, there would be DNA methylation of key and neuroendocrine traits on the right, and that wouldn't occur at low density. And then we would then measure this in the animals. So how are we going to test this? Key person was Phoebe Edwards. Phoebe Edwards and I, in the spring of 2016, built 8 enclosures. You have to realize there's this sort of passion that occurs in people like myself and Charlie and so on. I'm 61 when I'm hammering this in, all right? Obviously, Phoebe's a lot younger, but we work like crazy, trenching, building these enclosures so we can make essentially unique enclosures and manipulate density in each one of these. So here you can see the field enclosures built. On the top, you see we have 8 enclosures. They're essentially 25 meters by 25 meters. And on the bottom, you can see all the work that we did to keep predators out. We had electric fence, 7,000 volts going around it. We kept the voles inside each fence by having little aluminum barriers. They couldn't climb out. And all of this took place, and we replicated the study 3 times. So here's what we did. We had 8 enclosures. We used meadow voles, the one I've already talked about, and we created low density enclosures and high density. We knew what the density should be like because of all my trapping, which I did in nearby meadows. So these are realistic numbers. We got source animals from the surrounding meadows and seeded them into these enclosures in 3 different years. Okay, and this is the combined data of all 3 years. And what you can see, they're all introduced simultaneously in early May. They breed, have litter 1, population goes down. Portion of females lactating goes down. There's litter 2 and litter 3. The low density population does more reproduction than the high density population. Okay, so what are we going to look at? Well, again, it's based on the laboratory evidence such as the rat literature, but also the human, what we should look at. So we looked at two things: estrogen receptor alpha on the right side at the very top, and that one's key. Evidence indicates that that's key for parental behavior and aggressive behavior. So we thought it was a likely candidate. On the, I'm sorry, that was on the right. On the left, we're looking at the master regulator of reproduction. It's called GnRH. GnRH is produced in the hypothalamus. It's essentially a protein which affects, which migrates down to the anterior pituitary, causing it to release hormones which affect either the ovaries or the testes, and the animal breeds. All right. Okay, so our predictions were, Phoebe's predictions and mine, were that at low density on your left, there should be more reproduction, higher levels of ER, estrogen receptor alpha, and GnRH, gonadotropin releasing hormone. In the medigdala, there should be lower ER alpha. The flip side is in the high density populations, it should be inhibited reproduction, lower ER1 and GnRH in the hypothalamus, and higher in the medial amygdala. Okay, so what we did is we monitored these populations throughout the summer, maintaining the densities. And at the end of the summer, we collected youngsters that had been born in these two environments, collected young voles, brought them to the lab, euthanized them, collected their brains, and then sectioned them and extracted the tissues from the hippocampus, the paraventricular nucleus, and the amygdala. And then we analyzed for the methylation patterns in them. Okay, and so here you can see it. This is all done. It's a heck of a lot of work, but we then, here you can see the brains, the medivole, young medivole brain on the left. We extracted DNA from the key sites. We knew what to look for because we had the mouse atlas brain, and Phoebe extracted the tissue and then digested it through a whole bunch of steps. As I indicated, the problem that we had working on meadow-voles is that no one has sequenced the entire genome of the meadow-voles. However, in the prairie vole, Microtus ochragaster, an enormous effort has occurred because these things are monogamous. All other, essentially, voles are polygynous. The males only breed, and that's all they contribute. The ochragaster, the prairie vole, is unique, and a huge amount of effort has been placed in terms of sequencing the genes. The DNA is fairly similar to what we find in Microtus pennsylvanicus, the meadowvole. And Phoebe used that template to assess what the genetic structure and the methylation patterns were like in the meadowvole. But that was absolutely critical. Okay, so if you look simply at the expression of the gonadotropin releasing hormone, this is essentially the RNA. So this is not looking at the genes themselves, but it's looking at the RNA expression. And you ask the question, what does the RNA expression, which results ultimately in reproduction, what does it look like at high density and low density? What you can see is that at high density, the GnRH, which regulates reproduction, the master endocrine, was lower in juveniles born at high density. And that's what you expect. Okay, that's your red bars. The blue ones are the ones at low density. They're much higher. And that's basically what you would expect. If they're breeding more or going to breed, you would expect the GnRH expression to be higher if they're going to breed or about to breed. whereas the others wouldn't because they're at high density. Now here's the critical evidence. What Phoebe did is she looked at the genes themselves. She looked at the genes which the GnRH promoter, which determines whether the gene is expressed or not. So we knew the sequence, Phoebe did, knew the sequence of what the DNA looked like, the promoter section, in meadow voles. And the data is for males and females. If you look at the males, let me step back. She identified 6 sites. They're called CG sites. This is where methylation occurs if it's going to occur. All right? This is where it's going to occurs if it's going to occur. She identified 6 CG sites in the GnRH promoter. And then she looked whether they were methylated or not. If methylation occurs, essentially that gene, the gene which would be active, is inhibited from being active or less active. And what she found is in males, there's no difference. None of these methylation patterns in high and low density in the males is essentially no different. That's not the case for the females. For CP1 and CP2, they're on the, I always have to get they're on the right side. There are significant differences between the low density and the high density. As it says in the bottom, the females born at the high density had higher methylation levels at 2 critical sites. And this aligns with prior observations that females from high density environments retain reduced reproduction long-term. Again, going back, remember that at peak density, typically females, young females that are born that year, they're the only ones which will be around the next spring. They will retain this memory. And so the inheritance, the memory, as Mihawk and I showed in the 92 paper, will still be there. So you don't see that in the males, but you do see it in the females. So this essentially is consistent with the hypothesis. Okay, so this is basically the last slide, which summarizes this critical data. At low density, you've got, in this case, 2 females, and you have a large proportion of females lactating. Many of the young mature. I didn't show you this data, but their evidence of hormones in their feces are high. And what you see is the GnRH expression. The messenger RNA is high, and their promoter patterns is low, which means that they're reproducing. The flip side on the high density animals is that the proportion of females lactating are lower, the number of which mature are lower, and their fecal androgen levels, i.e. they're not reproducing, are also lower. We look at the bottom red, that essentially is a reflection of lower expression of essentially the protein, the mRNA, which is expected. But what is absolutely unique about this study is that GnRH promoter methylation is increased, which is exactly what we predict. Okay, so this is essentially the first line of evidence that shows that methylation patterns are occurring, that they are potentially explanatory for the voles and lemming cycles. Okay, and this study is just being published. As you can see, it's February the 6th. It's, I think, a really important study, incredibly important. It's a mechanism for population self-regulation. Social density suppresses GnRH expression and reduces reproductivity in voles. If you wish a copy, it's right now still, it's not the final copy, but you can find it online. Okay, so the conclusion is our results support a physiological basis for population self-regulation in vole cycles, as altering population density alone induces reproductive downregulation at the hypothalamic level. Our results demonstrate that altering early life social environment can fundamentally impact reproductive function, and that occurs through epigenetic changes at the GnRH gene. I didn't show the estrogen receptor alpha. We did not see changes there. And finally, this mechanism, either through social behavior, which is what we're showing here, this is what we're arguing is the cause, bulls and lemmings, or predator-induced stress in snowshoe hares, or similar processes in other wild mammals, may drive population demography. So that's our conclusion. The one thing I didn't say is with respect to the snowshoe hares, we are, with collaborations from sick kids in Toronto, sequencing, they've sequenced the DNA for snowshoe hares, so we intend to do the same types of things in snowshoe hares as what we've done. They have about 2.8 billion base pairs, and we're now annotating the genes, the critical genes. And so hopefully within a year or year and a half, we will have the story more comprehensively in snowshoe hares as well. So here's something to always remember, and I think it definitely speaks to where Dennis Chitty was, and where I've come. It says there are many things that hinder sure knowledge: the obscurity of the subject and the shortness of human life. Dennis didn't live to see this, but I think our findings and the findings in the future will say that, in fact, he was right. I'd like to thank all the people that made this study possible. Obviously, you see Charlie at the end there in the raincoat when he was younger. Phoebe was absolutely critical for the voles. Sophia Laverne is absolutely critical for the snowshoe hare. They work together in terms of trying to figure all of this out, because remember, they're ecologists, also endocrinologists, but this it's a steep hill to climb. And then we have the tracking team, and I thank them from the bottom of my heart.
Thank you very much, Rudy. Thank you for a great seminar. It's quite complex. It's amazing to see in just 1 hour what has been the work of a lifetime.
Yeah. And
the results you presented are a great contribution to this intrinsic population regulation ideas of Dennis. Dennis Chitty would be really proud of knowing these results, I'm pretty sure. It is also amazing to have this integration of ecology with neurophysiology and epigenetics. And there's a few questions here that I'd like to gather from the audience. There is one that is shared by Mariana Ferreira and Francisco Dorantes that has to do with interaction between behavior and food. And they say that what would happen if these experiments where when you have these females at high density were supplied either with quality, high quality, or with quantity of food, what would be the maternal effect? What would be the interaction between these epigenetics and the quality or quantity of food?
Very good question. So 2 lines of answer to that. Again, these guys are seasonal breeders. So as seasonal breeders, the breeding density is basically set at the onset of the breeding season. So, and so that's the first thing that one always has to bear in mind. And so again, depending on where you are in the Arctic or in Southern Ontario or in Indiana, the amount of food that's available is going to vary. There's obviously going to be a lot more food, so natural food. So the first thing is, what is the starting off density for the species in its natural habitat? And could you ameliorate the competitive interactions between females? And I think the answer is probably correct that you would. The ultimate question will be, what will this do to the progeny? So, and when are the stress effects going to occur? If you look at our situation in the Yukon. In the Yukon, we study, we've got a number of vole species, but the key one is the northern redback. And so they have much lower food levels than the animals here in Southern Ontario. Food experiments have been done there. Carlos has done those. We haven't looked at the epigenetics of that yet. But I think if it will ameliorate the negative interactions, in other words, if you supply abundant food at the peak density, could you make the peak density even higher? In other words, and that would not cause programming in the babies. I think it'll probably really rapidly increase in density so that the animals that are born in the late summer and midsummer are still going to experience that. But I think it may result ultimately that the young females may mature on site with their mother, so that whereas under normal densities, that would be much less probable at high density. So food could alter the trajectory, but I think it will essentially be temporary. It'd be worthwhile trying.
Okay. Another question is, do you think these maternal effects account for the cycle intervals? Like, are they responsible for maintaining the low? And how many generations could they last?
That's an incredibly good question. So the question you are basically getting at is, how long does this methylation last? How long, when does it come off? And there's basically two lines of evidence. I think the predominant one is that, but it's not absolutely certain because, again, it's based on rat literature from the lab. But one line of evidence suggests that when ova and sperm are produced in the next generation, the epigenetic tags come off. So essentially, that line of evidence argues that this maternal effect would last essentially one generation. So the females that are produced at high density would carry this signature, but if they were at all successful, or a subset of them were successful, they would then produce the increase. But the next generation, they would not be good quality animals, but it would essentially take a 1 or 2 generations before the epigenetic tags, the methylation patterns come off, and you're back to square one, where they're really good quality. But let me step back. Michael Sheriff, this is on the Snowshoe Hares. He asked a very simple question, and we looked at the length of the low in Snowshoe Hares, and you may look at it that the length of the low varies. So the low that we saw, in the mid 80s was almost 5 years, where the length of some of the more recent lows, or some of the lows that Lloyd Keith had seen, are less. And what Michael tried to do, and it was an indirect approach, is the length of the low related to the intensity or the speed of the decline? And that's what he showed. I mean, it's only based on the sample size, really hard one. I think of about 6 low phases, but the more intense the decline, the longer the lows, arguing that under extreme predation risk, the methylation patterns are much more pronounced. And and again, we're only looking at GnRH and GR, but it's highly unlikely, like you saw in the suicide victims, that it's just those genes. It's probably a whole suite of genes. We can't answer that question yet, but that's what's probably going on. Anyways, that's entirely a hypothesis. So in other words, how long is it going to last? The lab evidence said it should last maybe 1 or 2 generations. That's it. Some of the fields suggest it lasts longer, but we're looking at natural populations, not the lab.
One question, I guess this is from my own curiosity. there are species that don't have cycles, or at least don't have strong cycles. They have annual cycles, low densities in the mammals, mainly granivores. All these Peromyscus or these field mice that eat seeds are very stable. I was impressed the time I studied deer mouse. I was very impressed by the stability of their populations. And has there been any similar work to what you are presenting here on populations that don't cycle? And do you think that these mechanisms are going to apply to them too?
So, to my knowledge, there is no studies which have looked at populations which are essentially stable over time with respect to epigenetic changes. Okay, I don't know of it. And so then the question is does it occur there? I think you could extend that argument with respect to, say, the ungulates who are challenged on a regular basis by, in our case, in North America, by wolves, etc. In the case of the Serengeti, by lions and things like this. Would you expect epigenetic signatures to occur there? And I would predict that you wouldn't expect it. In other words, it would be a function of the evolutionary history of the beast. That for whatever reason, the intense social structure in hares, in voles and lemmings produces this. For animals like the deer mouse, you would predict that it would not occur. It would not be a population-wide, because here what you've got is a population-wide phenomenon. Everybody does it. And so they all go up through these cycles. You would not predict that it would not be population-wide. There may be individuals which are better or worse than others, and you would be able to predict that out. But the population itself essentially remains constant. So my expectation would be you would not see this population-wide methylation pattern that I would expect you would see in voles and lemmings. And in some populations, such as those which are stable over time, say the large ungulates, I would not expect it occurring there at all, even with predator threat.
Okay. And one final question, Rudy, since you like to take examples from humans too, Daniel Dulcin asks if you can give your insights on human population evolution during the coming decades. I guess since you teach the biological effects of climate change, you might be able to.
Well, I think that's a really good question as well. And I think you can ask the question, if we look at the past and the evidence that we have in human populations with respect to how mothers had children and so on, what were the potential consequences of living under war or living under starvation and things like this? Are there methylation? Is that predicted? And I would argue that it is. So when we were, Carlos and I were talking before we started about where I came from. I come from the Netherlands. And my parents and most of my siblings, my brothers and sisters, went through the war. But we were in a province which had food. In 1944, it was called the Dutch Hunger Winter. One of the major workers, Crutzen, just died, climate change. He went through the Dutch Hunger Winter. The question is, what are the implications of that? What we're looking at women who in Amsterdam, Rotterdam, and The Hague, but they were essentially starved. What were the impact on their babies? Many of them lost their kids, but for those that survived, they essentially affected up to 2 generations. It affected the daughters and then the daughters of those daughters. So if you would say, what will happen? I mean, obviously, that would be hunger, severe stress because of war. I was just talking to one of my friends. She works in the cardiac unit at the Toronto General. And she's noticing that young people are becoming extremely stressed because of job loss and things like this. So you can see that all of these things could affect the behavior and potentially the epigenetics. What I focused on in my examples is the epigenetic changes in young animals. But the human example looked at children. It's highly likely that epigenetic changes are occurring throughout your life because of your experience. So it's not, you know, you've got your DNA and that's what you're saddled with, and nothing's going to change. Because of severe stress, it's highly likely that those stressors will be written onto your DNA and epigenetics, and yet you're different after those experiences than you were before. So you're a different human being. So I suspect that, you know, depending on where you live, I live in Canada, but, you know, if you're living under severe dictatorships, if you're being exiled, under persecution, and you can think of the countries where this is occurring today, they're under a lot of duress. And those signatures will be written on those human beings, without question.
Well, Rudy, thank you very much for a great talk. It's been a great pleasure to listen to this amazing story and to see the new insights into the intrinsic population regulation. Thanks again, and well, we'll continue with the seminar series. Thank you, everybody. I'd like to tell you that we had people from many places, about 20 states from Mexico, from the north to the south, but we also had people from Brazil, from Uruguay, from El Salvador, from Washington, DC. Thank you, everyone, for watching, and see you next Thursday here.
Excellent.
Bye-bye.